Good afternoon, everyone, and welcome to the pharmacology class. And uh, today's uh, topic is anti-fungal drugs, uh, and the fungi are very different, not very different from the human cells. And if we look at the classification, fungi are in the same domain as the animalia and human. So fungi are actually eukaryotic organisms, and they are multicellular and do not photosynthesize. Their cell walls are different from other organisms, including the humans, in that they have a high tail. And also, the main function of the fungi is absorption of nutrients and decomposing the organic material. So it can be used further for the photosynthesis by the plants uh, and animals. So the classification of the fungi is based on the uh, type of the cells, and so the two main families include the yeast and molds. So the yeast usually produce like a Saccharomyces cerevisiae is the yeast that makes bread uh, to be spongy, and then the molds uh, are another family that actually can be used in some French cheese to make it tasty, but also both of these families have a different pathogenic fungi that include the Candida albicans and other species, Dermacytis giravensi that mostly is seen in HIV patients, and also the uh, molds include the Aspergillus species that is very resistant for the most of the uh, and, uh, anti-fungi drugs, and also mucormycosis. This is the classification of the fungi, and the fungi are actually the uh, have a different cell wall, and uh, this is the cell wall of the fungi that is mostly composed of the proteins, minor proteins, and beta glucans, and also chitin that makes the cell wall very rigid, and that helps to protect from the bacteria. And also the plasma membrane of the fungi consists of the phospholipids, bilayer of the phospholipids that also have an ergosterol as the primary agent to make the membrane elastic, and this is different from the human cells that have cholesterol instead. So and because of that, most of the um, anti-fungal drugs are targeted against the ergosterol production or synthesis. So, and one of the first drugs uh, for the treatment of the fungal diseases is amphotericin B that is acting on ergosterol and making pores that allow the leakage of the ions from the fungi cell, and that leads to the death of the fungi. Uh, and but unfortunately, amphotericin B is non-specific, and it also can make pores from the cholesterol molecules as well, and that leads to the high toxicity, and that is why nowadays amphotericin B is only used in severe conditions that require emergency treatment, and so usually are used intravenously. So amphotericin B is very toxic, and is mostly toxic for the cells of the uh, uh, kidney. So, and the kidney usually uh, uh, will be affected by the amphotericin B. That is why uh, mostly contraindicated in uh, patients with kidney disease. Uh, another another way of acting on the ergosterol is removing preventing the synthesis of the ergosterol, and in fungi, ergosterol is synthesized from the squalene with the help of the squalene epoxidase that is used to make the lanosterol, and that is converted to the ergosterol by the 14 alpha demethylase. So, and these steps are inhibited by the two uh, two uh, two different chemically structured different drugs, like as allyl amines, and allyl amines include naphthene and terbinafine, and the derivative of the allyl amines butenafine. And the second step is inhibited by the azole derivatives, including the imidazole and also the 3-azole derivatives, and that includes the fluconazole, and also clotrimazole, ketoconazole, miconazole, and voriconazole. So all of these will decrease the synthesis of the ergosterol, and that will make the membrane non-elastic, and that will lead to the death. Unfortunately, these drugs also inhibit the enzymes of the cytochrome 450 in the liver, and that is why they are toxic to the liver. So that is why they are not very safe. The third way of inhibiting the cell wall is the inhibiting of the beta-1,3-glucan synthase, that is an enzyme of the membrane, and the class of the drugs that is called echinocandins will inhibit this enzyme, and that includes fungi, uh, Caspo, Fungin, and Micafungin. And these drugs are relatively more selective because beta-1,3-glucan synthase is only present in fungi cells, and that is why there are less adverse effects and more selectivity, and also they are very effective against the Candida and Aspergillus uh, uh, families of the fungi, and that is why they are mostly used for the Candida uh, infections. And uh, uh, besides the acting on the cell wall synthesis, the uh, anti-fungi agents can uh, disrupt the mitosis, like glycofulvin, that is disrupting the mitotic uh, uh, division of the cell, but disrupting the spindle, and also uh, flucytosine uh, is another has another mechanism of action by converting to 5-fluorouracil and acting on the uh, nucleus of the fungi by preventing the synthesis of RNA and DNA. So these are the mechanisms of action of major uh, uh, clinically uh, vital anti-fungal drugs. Yes, and also the nystatin actually has the same mechanism of action as amphotericin B, but because of the toxicity, nystatin is only used topically for the treatment of the skin infections and mucosal infections, like the infections of the uh, 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 mouth and vaginal infections. Uh, so and also and some enteric infections, so it's not actually uh, absorbed from the intestine. So these are the major clinically effective uh, anti-fungal drugs, and so their mechanisms of action. Another classification is based on the types of the diseases in which the drugs are used. So all fungi infections can be divided to systemic and cutaneous. And for the systemic, we mostly use those drugs that uh, can be absorbed or can uh, distribute to the all tissues of the body, and that includes amphotericin B, flucytosine, uh, and also as triazole antifungals. So azole fungus that are imidazoles are used mostly for the cutaneous infections, but triazole antifungals are used for a systemic, and they have a conazole ending. Like fluconazole, itraconazole, posaconazole, and voriconazole, and then also echinocandins are used also as a systemic uh, drugs. And cutaneous include the drugs for the treatment of the skin infections mostly, and this includes covalent epoxidase inhibitors that end with the phen, like terbinafine, naftifine, and butanafine, and also the toluene that disrupts the mitosis, nystatin, same as the amphotericin B for the mechanism of action, but only used for cutaneous infections only. And then imidazoles include clotrimazole, ketoconazole, and miconazole, and also cyclopirox and toinaftate are used as only cutaneous infections. And the mechanism of action of amphotericin B is based on the formation of the pore from the ergosterol. So these are molecules of ergosterol, and the amphotericin B will make a pore that leads to the leak. And amphotericin B is very toxic and can be injected intravenously and also used topically. It's eliminated by the bile and urine, and the adverse effects include high temperature, chills, kidney failure because of the toxicity for the kidney, and that because they are non-selective. Hypertension and anemia can also be seen in some patients. So for that reason, amphotericin B is no longer used frequently and only in emergency cases, and also sometimes topically for the severe infections. Fluconazole is inhibiting the ergosterol production by inhibiting the enzymes, and that is more safe way, and that is why fluconazole is commonly used for the treatment of the vulvovaginal infections, vulvovaginal fungi infections, usually by candida, and that is the most common indication for the fluconazole. And all of the triazyl derivatives may lead to because they disrupt the metabolism in the liver. They can interfere with other drugs like that are also metabolized by the enzymes of the liver. So that is why these drugs can increase in the concentration and lead to the toxicity. For example, warfarin will increase the bleeding. Phenytoin may lead to the seizures, and cyclosporin and triazolam can actually lead to the increase in the adverse effects. And the squalene epoxidase inhibitors do the same, like they also inhibit ergosterol synthesis, and that increases the concentration of the squalene in the fungi cell, and squalene itself is very toxic for the fungi cells, and that is why that leads to the cell death. So terbinafine and naftifine are very toxic. That is why they are very toxic for the fungi cells, and they can be effective orally and excreted by the urine. And greaser fulvine actually blocks the mitotic spindle formation and leads to the inhibition of the division. That is why it is fungistatic, and usually is used for the treatment of severe conditions because of the non-selectivity. So it also disrupts the mitosis in other cells as well. So I think so actually the fungi are fungi are playing a vital role uh, in the world because they are not only causing diseases, but they also help the plants to absorb the phosphates that increases the crops, and also we use fungi to produce a beer and wine, but also uh, to make a bread, and many research that was done uh, like 100 years ago, for example by Pasteur, is used. So like uh, when Pasteur was working in France, he uh, he researched on the beer production, and the government gave him a goal to increase the beer uh, life so it can be preserved longer, and then Pasteur started to look at the uh, fungi under the microscope, and uh, uh, he have found that fungi are living cells, and besides the fungi, he also noted another microorganisms, and the further research led to the result that by heating the beer uh, at the up to the temperature of 50 degrees can kill all the bacteria, and prolong the life of the beer up to several months, and that is called pasteurization. And further uh, has been used for the other products like dairy products like milk, and like today we know like pasteurized milk has long life, and also recently they invented ultra pasteurization that uh, can heat the milk up to 140 degrees by Celsius, and also for the several seconds, and that makes long milk even longer life, shelf life like up to half year. um, and that help to preserve the food longer. So uh, and some of the fungi are uh, can be toxic and lead to the death, but most of them are very important for the ecology because they decompose uh, the dead animals and plants, and they can decompose the polysaccharides that is very important uh, for the uh, for the ecology, and oh, so and there are not so much diseases like uh, that are caused by the fungi, but and most of them are not fatal diseases. So and some like most of the candida diseases, like candida is semi-pathogenic, so it's normal microflora for the vagina, for example, and normally its growth is suppressed by the bacterial flora. But when the bacterial flora is inhibited or is removed by the antibiotic, for example, uh, that is ineffective against the fungi, then the fungi can grow further and lead to the disease like the Candida disease. But normally it's like normal microflora. So I think that is it for today. Do you have any questions?
No, thank you.
No questions. So for like for the vulvar vagina, for the vulvar vaginal infection, the most common drug used is fluconazole. It's very easy, so like only one tablet is required, and it's taken orally, and it's very effective against the Candida albicans. So the same is can be used for the thrush in the mouth and other Candida diseases that are most common. Okay, then thank you for your attention, and see you next week. Bye bye.
We will have no class?